All right, shall we get started then? I think we have a good number of people. There might be some more trickling in, but welcome everyone to our VMOL seminar. We're very excited to be here with our special guest speaker, Simone Zuffa. And just a bit of background on Simone. So he did his undergrad in Bologna. He's from Italy. Then he moved to do a master's at Wageningen, which I can't pronounce the name of that so well. He did his master's in medical biotechnology, and there he was actually doing anaerobic microbiology. He was a wet lab scientist at the time. But then in his PhD, he sort of switched. He had a passion for data science and computation. And so he did his PhD at Imperial College in clinical research medicine. And from there, he wanted to dive deeper into metabolomics, moving to UCSD to do his postdoc in Peter Dorostein's lab. And that's how Daniel, Simone, and I all met each other. And so we're quite excited today to have Simone talking to us about some tools for actually functionally annotating and contextualizing MS spectra. Before Simone also has a passion for traveling and skiing as well. And we got to experience that last weekend. He was actually visiting Denver for just a few 30 hours. So always great to have Simone. And with that, take it away.
Okay, thank you so much, Allegra. Let me share my screen. Okay. You can see my screen, right? So, okay, thank you so much again. I'm here today to introduce you to a tool that we have been developing in our lab at UCSD in the past 1 or 2 years called MicrobeMASST that hopefully will be help you making sense of your MS data in the context of bacterial, fungal, and other microbial metabolites. So, as Allegra just mentioned, maybe I'm more oriented towards the clinical medicine and clinical metabolomics side of this field. And I'm really interested about how metabolomics can provide a proper readout of the phenotype that we observe in specific diseases. You were probably very familiar with this, but usually it works like this. You pick your favorite and most horrible diseases affecting the most innocent people. You collect all this kind of biosample like blood, stool, urines, and then you run them through your favorite instrument that can be either a mass spec or an NMR. And then you obtain a fingerprint of the molecules that are observed in these particular samples. And then you try to make sense of all the data that you have collected, and hopefully it will also make sense in the context of your health and disease question. The metabolomics is a very hot topic that usually goes hand in hand with another very hot topic, the one of the gut microbiota. Hopefully some of you are familiar with that, but we have literally trillions of microorganisms that are colonizing our body. And this bacteria and other microorganism produce like 100 or 1,000 of molecules that can reach every organ in our body, and then they can influence our metabolism. These bacteria can produce very important energy metabolites, neurotransmitter, vitamins, but what is more interesting that they are probably are producing many more unknown molecules for which we have no idea of the function yet. A nice example that I want to bring to you is like two different molecules with two completely different function. One is an anti-inflammatory molecule and another one is a pro-inflammatory one. So the first one, indole 3-lactate, that we know is produced by Bifidobacterium species, was shown to reduce inflammation through the regulation of T helper cells. While on the other hand, colibactin, that was shown to be produced by Enterobacteriaceae, has been also shown to be involved in inflammatory bowel disease and colorectal cancer insurgence. So we can see that there are two completely different action of that microbial molecules can affect our body. So how do we study the microbial functionality? Well, in our group, we mainly use mass spectrometer, and this field can actually be divided in two different branches. One is a more targeted approach that is used for hypothesis testing and quantification, but the main drawback is that it's mainly based on primary metabolites and commercial available standards. In our group, we are more focused in the untargeted metabolomics that is focusing on secondary metabolites, so microbial metabolites, that is though more involved into hypothesis generation and aim at capture all the molecules that are possibly observed in a specific sample. But there is a big drawback in the untargeted approach. We have almost no idea of what we are observing, and this is the dark side of untargeted metabolomics. The annotation rate that we are able to obtain from the MSMS that we are acquiring is increasing through the year. But what is very interesting is that within these annotated molecules or MSMS spectra, just a very tiny fraction is actually of exclusively of microbial origin. And this doesn't reflect at all the enormous genetic potential of the microbiome that encodes 100 times more gene than the human genome. So there is a lot of potential there and a lot that is awaiting to be discovered. So how can we make sense of all the molecules that we are actually capturing through our studies, since the annotated part is a really small fraction? Well, we know that all the molecules that we are capturing are somehow coming from either the environment, the food we eat, the drug we take, and also by the bacteria that inhabit our body and then they produce these metabolites. This task of making sense of all this mess seems very daunting, but luckily for us, the community is constantly depositing metabolomics data that we can start leverage to make sense of what we observe in our studies. So for this tool, we built MicroBus that is basically a search tool that can compare the MSMS that you observe in your study, hopefully coming from bacteria, to data that was already deposited in the public repository of metabolomics data. And the main advantage of this tool is that it can be agnostic. So you don't have to use an annotated MSMS spectrum, but you can use also a non-annotated one that you have no idea of what it is. But this tool will be able to point you to possible producer of both known molecules when you search for them, or a known molecule. And this is the only tool that can do it at the moment. So as Allegra mentioned, before the MicroBus paper was actually just recently published last week, and we are very excited about it and the buzz that has generated in the community. And so today I'll just give you a brief example and story of how we build it, how it works, and what are the limitations. So how do we build MicroBus? We started by scraping all the public repository data that we have in the GMPS massive ecosystem. For the one not familiar with that, there are mainly three metabolomics repository: metabolites, metabolomics workbench, and GMPS massive. So we focused on the one that we mainly curated. We both curated data manually, but also through Redo, that is another tool developed in our lab, which allows user to associate to their sample vocabulary controlled metadata. So then we turn to Peter, that is our in-house Twitter star, to ask the community to deposit even more data. And the response was like astonishing. In just 2 months, we had 25 different research groups all over the world deposited their data, and we ended up obtaining for this specific study more than 60,000 samples comprising 100 million of MSMS spectra collected either by monoculture of bacteria and fungi or human cell lines and QC and blanks. So we have a lot of data, but a lot of data is useless if you cannot quickly search through it. So luckily for us, Ming Wang, a former postdoc in our lab, now assistant professor at UC Riverside, was developing a fast search tool that basically allow you to index all the MSMS spectra present in your repository that you have curated, and allow also to search your spectra of interest against this repository in just seconds. And this was like a major advancement compared to the tools that previously before, because it was taking for around 20 minutes to search for one single MS spectrum against our repository. So now we have a lot of data and we can quickly search through it. What we needed next was a structure. So as I mentioned before, we had 60,000 Unix files coming from monocultures of bacteria, fungi, and archaea. We decided also to add human cell lines and QC and blanks in order to use them as negative controls when you were doing your searches. And with the metadata that we were able to find and that was associated with this file, we were able to extract more than 2,000 unique NCBI IDs that basically are IDs that just represent a specific taxa in the ecological world. We decided to construct a taxonomic tree instead of phylogenetic tree because all the samples that we were able to collect did not have associated genomic data, so we were not able to compare them genomically in an accurate way. Also, some of them were novel strain that were not yet deposited in CBI, and we also had to overcome the fact that we had to generate a multi-domain tree. So using the NCBI IDs, was that we were able to reconstruct around 2,000 unique lineage that were then just collapsed into a taxonomic tree of almost 3,000 nodes. This taxonomic tree spans across 3 domains of life. You can see there are more than 16 phyla, and if you are a bit familiar with microbiology, you will recognize a lot of them. And in our reference database, we have bacteria that were isolated from the environment and for human microbiome. So we kind of cover most of the hot fields in the microbiome world. So basically that's it. MicroBus is a tool that allows you to search quickly an MSMS spectrum of interest against indexed reference database, and then allows you to visualize everything in an interactive taxonomy tree. Robin Schmidt, was also a former postdoc in our group and now doing amazing work in Prague, put everything together in a very user-friendly web interface that you can explore already at the GMPS2 MicroBMASST website. So I'll quickly show you a live demo of how it works. When you open the web page, you will see this interface. You will be able to search for your MSMS spectrum either by inputting a universal spectrum identifier that, if you are not familiar with that, is basically the equivalent of a URL, but for MSMS data that has been deposited in a public dataset. Or you can just go more traditional and copy paste your MSMS spectrum in this window. Then you just have to define some of like search tolerances, and then you just have to click search by MicroBMASST in case by USI in case you have inputted a USI. In this case, I'm using like a library spectrum in the GMPS library to showcase the search. So once you click search, in just like a second, you will be able to obtain the interactive taxonomy tree. You can see, you can explore it. You will see that there are different pie charts. This pie chart represent the number of samples in the specific reference database that have found to be a match to the specific MSMS that you've searched for. You can see there are scientific names, the NCBI IDs, the genera or phylum that you were looking for. You can collapse at the different level from phylum to strains, the tree. Also, along with the tree, will be generated 3 different tabs. The library tabs show you the matchings to our library datasets. In this case, we have a perfect match to Yersinia bactin because I searched for Yersinia bactin spectrum. In the dataset match, you will be able to see in which sample and which scan the MSMS that you searched was found to match. And we will also be able to open a mirror plot that will show you exactly the match. In this case, you can see in the top, we have the library that I searched for, and in the bottom, the spectrum from the samples, and there is a almost perfect match. Finally, in the taxa match, you will see all the taxa at the different levels and the number of matches and the fraction of samples that were found to be matched compared to the total. So, and basically this is just the user interface that you can explore. Well, okay. Um, what we are really proud of is that MicroBus is very accurate because, for example, when we search for medical molecules that are medically interesting, like lovastatin, that is a cholesterol-lowering drug that was originally isolated from Aspergillus, and salinosporamide A, that is a phase 3 candidate to treat glioblastoma, that is a brain cancer that was also produced only by Salinotropica. We can see that this MSMS spectrum were also only found to match to what we were originally isolated and what we were expecting. So in the in the paper, if you had the chance to give it a read, I showcase how we can actually extract and use Microbus to explore potential microbial metabolites that were never discussed before. So to do that, I showcase how we can take an animal study. I took like the Queen data sets that maybe some of you are familiar with. It was published in Nature a couple of years ago, where germ-free mice were compared to specific pathogen-free mice, so mice that were colonized with a microbiome. I was able to extract all the MSMS spectra from these samples, almost 2 million MSMS, that were actually around 30,000 unique ones. And I was able to extract only the MSMS that were observed in the animal that were colonized with bacteria. Once I have extracted all these MS, around 10,000 unique MSMS, I was able to search them all through MicroMASST. And 10,000 of them were found to have a match to at least one of the bacterial monoculture that are present in our reference database. To refine this search, I then removed all the SMS that were also found matching human cell lines because we wanted just to refine and focus only on truly uniquely microbial metabolites. And then I validated this finding against another antibiotic, another study where mice were treated or not treated with antibiotic. And I kept only spectrum that were observed in the mice that were not treated with antibiotic. And at the end, I end up with around 500 MSMS. So this data, all this data that has been generated, there's MGF files with all the list of these MSMS are actually available for you to download them. You can also see that these 500 spectra were actually fairly distributed across the phyla. You can see that some of them were actually present in bacteria coming from all the different phyla. It was so interesting that, for example, terpenoids seems to be more universally shared across phyla, while alkaloids and amino acid peptides seems to be actually more specific to the different phyla. So, as I mentioned before, though, I'm interested in clinical medicine research. So the next step was to search these 500 spectra against all human data that is deposited in the GMPS massive repository. So I was able to find 550 of them matching sample acquired from different location of the human body. You can see blood, you can see stools, obviously, across different diseases and in healthy patients. And finally, on the figure on the right, you can also see the number of matches that were constantly found. Obviously, stool had a lot of matches because we mainly focus on working on microbial metabolites presence in stools. So these also recapitulate and showcase of how microbial metabolites are actually not restrained to a specific organ like the intestine, but they can be found all over the body. I also want to showcase another application. For example, when we search for a new conjugated bile acid, that hopefully maybe you have read the paper that was finally published in Nature from our lab a couple of weeks ago too, phenylalanine acid, that we showcase to be increased in presence in individuals that are affected with inflammatory bowel disease. If we search for the SMS spectrum of this particular conjugated bile acid, we will able to observe it, to match monocultures of bacteria that were previously actually associated with IBD. Obviously, this can help a microbiology scientist and people working in the microbiome world to pinpoint and validate also the result, because one of the big drawback of microbiome research is that it's not important just what bacteria are there, but actually what is their function. Another thing that is also very interesting to notice is that all this data was actually acquired years before phenylglycolic acid was observed. So these tools also allow you to retrospectively study your investigate other projects that were previously acquired. Finally, I want to just discuss about a bit of the limitation and the future developments. So regarding the limitation, obviously a match to this bacteria means that they are possible producer, but if you don't find a match, for example, to Akkermansia, it doesn't mean exactly that Akkermansia is not able to produce this molecule, but only that maybe the condition that Akkermansia was growing, or the methods that were used to extract the metabolites for that specific study did not allow us to observe that specific metabolite. So this is the main drawback of limitation. But a lot of other questions that people ask me is that, but what about community metabolism? Because we know that bacteria can behave very differently when they are in communities. Another big question is like, what about unculturable bacteria metabolism? Because maybe you are not familiar with that, but most of the bacteria that colonize our gut cannot actually be cultured with traditional technique that we use today. Other question also involved, what about association between observed microbial metabolites and diseases, or what about the biolocalization of these metabolites? Can we somehow investigate that? So we are developing something new in the group, and we started interrogating all the unstructured data that we have in our repository. So as I mentioned before, GMPS Massive, it's the repository that we use the most for our metabolomics study, where we deposit all the data. At the moment, we have almost 3,000 datasets deposited in Massive, and only 800 of them are actually being used to generate domain-specific masks that I've described to you. For the moment, I have described you only Microbus, but we have other domain masks called FoodMast, and we are developing a very similar tool to Microbus called PlantMast, where we use as a reference database all the plants that have been deposited in our dataset. So people with different interests will be able to use these different tools. As I mentioned before, we have Redo, that is a very powerful tool that allows you to associate metadata to the samples that you have been deposited. And on Redo at the moment, we have 75,000 files coming from almost 250 unique data sets. So I digged into this data, I did some filtering, so I removed all the overlapping data with the domain MAST and with the Redo metadata associated, and then I focused to retrieve only organisms, complex one, coming from the core data. For example, these involve primates, humans, obviously, but dogs, fish, we have a lot of different samples, and obviously also mice and rats. So we end up having almost 32,000 unique files, a CMS file coming from almost 90 datasets, actually spanning almost 1,500 NCBI IDs. So how does this data look? Obviously, a lot of the data that is deposited in our repository is coming from human samples, because we are focusing a lot on the relation of human health and microbiome, for example. But there is a huge amount of sample also coming from other model organisms like mice and rats. We have also almost 1,200 samples acquired from different taxas or from a gallbladder study. So that would be also very interesting to investigate. So regarding the tissue distribution, obviously, as I mentioned before, we have a lot of fecal samples because we are focusing on that, but we have also a lot of like skin samples, blood samples, or tissue samples, for example, brain and heart. As you can see, we have a lot of different tissue with different level of granularity. At the end, we also have a lot of different diseases represented. Obviously, the most represented sample are from people that do not report any disease, and these are usually the healthy control of your study. But then we have a lot of diseases represented that ranges from obesity, HIV, IBD, and for example, also COVID-19 samples. So the idea was to put all this information and construct a new structure, metadata-oriented tree. So this is a proof of concept that we've been just working couple in the last weeks actually. So I'll just to preview how we build it. So it's another tree that has been generated on 4 different levels using the metadata that was that we retrieved from Redo. And at the first level, we have a biolocalization, then we have a disease level, then we have a taxa, and then we have a sex. So the concept itself is basically that if you are doing your microbiome metabolomic study, let's say, and you find your biomarker of interest, you can search the SMS spectrum. And let's say that this biomarker was found to be associated with Alzheimer's disease. Now you will be able to see if this specific MSMS spectrum was also observed in other Alzheimer patients. And for example, if they were male or female, and where actually were localized, maybe just in the blood or just in the brain. So obviously we haven't solved Alzheimer problem. So I'll show you a quick example of an easy example of how this other tool will work. Obviously drugs, it's something that are not universally present in monoculture of bacteria or plants. So if we search for this specific antibiotic, azithromycin, that is mainly used for treating bronchitis and pneumonia, you can see that it finds a match to several different tissue in our database. And specifically, you can see that it is mainly found in blood samples, because usually azithromycin is actually an intravenous, is given intravenous to the patients. And you can see that half of the patients in a data set that was looking at people with Kawasaki disease, that is a non-communicable disease that mainly affects kids, half of them were receiving this antibiotic. And this makes sense because these kids were treated and were actually hospitalized. So you can see that we can easily retrieve the fact that we can find drugs, for example, antibiotics, through the human samples. This hopefully will inform you for your also for your study. So as a take-home message, the dark world of metabolomics and target metabolomics is very scary. But through annotation and hopefully through structured metadata, we can start making a sense out of it. Through MicroBus, hopefully, we will be able to help you to pinpoint specific known or unknown molecules to specific bacteria, and hopefully help you then to design more mechanistic study to pinpoint the metabolism of your bacteria of interest or of your molecule of interest. Um, Obviously, it takes a village to do this kind of jobs. I was very lucky to be in the Dorsing group that we all work together. Specifically, I have to thank Robin Schmidt that he was fundamental and is obviously one as the first co-author with me on this project. Annalise too, and obviously all the other people in the Dorstein lab that helped us on this specific project. And also all the people that deposited data for the reference MicrobeMASST database, because I forgot to mention, but we had almost like 100 people in the paper involved in this project that deposit data from all over the world. And that's it. So this was a quickly introduction to Microbus. So if you have any question, please feel free to ask.
Thank you so much, Simone. So we can give a round of applause, and perhaps now those of you that have to leave can step out, and those of you that want to stay for the Q&A can do so. And I already see some questions in the chat, actually, from Emmanuel. So